Hi, everyone. We can see that some of you have already joined, but we'll give everybody a few more seconds, maybe half a minute or so to continue to join, and then we'll begin. Thank you. Okay, I think we'll get started. Hello, everyone, and thank you for joining us. My name is Sushmita, and I work within the Inscopix marketing team, and we're very excited to have you all be here. So, for those of us who don't know us, Inscopix, we are a neuroscience tech company based out of Palo Alto, California, and we provide complete miniature microscopy solutions to neuroscientists around the world for the mapping of brain circuits in freely behaving animals. Today's webinar is specifically focused on our latest offering that we're calling the MIRA platform, MIRA being an acronym for Multimodal Image Registration and Analysis. Alice Stamatakis, who's here with us, is our senior lead scientist for application science and will be presenting today's webinar and answering all of your questions on our exciting new product. So just a quick introduction of Alice to you. She received her PhD in neurobiology from the University of North Carolina, where she studied the neural circuitry underlying motivated behaviors. She joined Inscopix in 2014 and since has driven product and application development for our company. Before we get started, I just want to make sure that everyone's aware of how to engage with us. So we ask that you please provide your questions via the Q&A box, and we will do our best to prioritize those questions so that Alice can answer them at the end of her presentation. If at all we are unable to address any or all of the questions, please know that it is only due to a lack of time, since we do have a hard stop at 10 a.m. Pacific. And with that, Alice, I will turn this over to you.
Great, thank you, Sushmita. So today I will be talking about our newest platform, which, as Sushmita introduced, is the Multimodal Image Registration and Analysis, or MIRA for short. So I'll actually start by giving a little bit of background on Inscopix for those of you that may be unfamiliar with our company and our technology. So I'll start by going over our two Miniscope platforms, the Envista system, which enables calcium imaging in freely behaving animals, and the Invoke system, which enables simultaneous calcium imaging with full field of view optogenetic manipulation. And then I'll spend the rest of the time going over the MIRA platform, which enables the registration of free behavior imaging data with high-resolution benchtop microscopy data. So as we know in neuroscience, there is multiple levels of investigation, starting from the microscale, where we really want to understand the role of individual neurons, through the mesoscale, which it deals with neural circuits, up through macroscale, which is where we start to look at brain regions. And at Inscopix, we were really interested in creating technology that could address this mesoscale level of investigation, because we really want to understand how neural circuits are related to behavior. So to enable that goal, we built out a Miniscope platform with the Envista and Invoke microscopes. So the Envista microscope is a single-channel epifluorescence imaging microscope. As you can see here by the dimensions, 8 by 15 by 22 millimeters, less than 2 grams, It's very miniaturized, it's tiny, it's small enough and light enough for mice to wear these mini scopes on their head and engage in naturalistic behavior. The excitation LED and emission are optimized for GCamp imaging. So we are using calcium activity to look at neural circuit dynamics. Our Invoke microscope, shown here on the right, is our platform that adds on optical components that enables simultaneous optogenetic modulation within the same field of view as the calcium imaging. So I next wanted to go over a typical workflow to do these Inscopix miniscope experiments. So the first step is to infect a particular brain region and cell type with the GCamp indicator. So we typically do this by a virus injection. Once the GCamp is expressed in our cell type of interest, we then want to enable optical access to that brain region. So we do this by inserting a grid lens that's attached to a base plate that allows the microscope to dock on and off and enables longitudinal imaging and allows us to come back to the same exact field of view over multiple recording sessions. So here's an example of what this imaging looks like. This was actually one of the first images from our first model in Vista. This animal is engaged in an open field behavior, and we're looking at hippocampal neurons on the right. So as you can see, this miniscope allows for naturalistic behavior. You can see that this is cellular resolution calcium imaging. The grid lenses that I mentioned in the workflow, we have these in a number of different sizes. So this really enables us to access any region of the mouse brain and many regions of the brains of higher order species as well. And because we can get the spatial information of these cells, this enables us to track individual neurons over time, which is a really great way to see how these neural circuits respond to different behaviors and how they are changing over time. So we next wanted to highlight a couple of research studies that were enabled by the Envista technology. So the first type of research that a lot of our customers engage with is functional cell type mapping. So this is where we have a specific cell type of interest, and we want to see how those cells respond to a particular behavior. So an example of this is from Garrett Stuber's lab. Here they were imaging from GABAergic neurons in the lateral hypothalamus as a mouse was engaged in this reward-seeking task where the mouse had to nose poke for a food reward, a liquid reward. And so there's two aspects of this task that they were tracking. First was the nose poke aspect of the task, so looking at the reward-seeking portion of the task. And then the second was the reward consumption, so when the animal is actually consuming the reward. And so if you look on the cell map here on the right, they identified cells that were excited by the nose poke portion, so cells that were more involved in reward seeking. These are shown in yellow. And then they identified a separate population of cells shown here in blue that were excited by the reward consumption aspect of the task. So this was an interesting finding because these, again, were all homogeneous GABAergic neurons within the lateral hypothalamus, but they actually started to see these different neuronal ensembles within this homogeneous mixture of cells. So this really underscores the necessity to start imaging these neural circuits at the cellular level. So another type of research that's enabled by the Envista technology is neural coding and decoding. So an example of this was from David Anderson's group. Here they were imaging in the ventral medial hypothalamus in male mice that were located in their home cage. And what they did is they introduced these mice to female or male intruders, and they imaged the neural circuit as the mice engaged with these intruders. And so what you can see in the cell map here are blue cells that responded to the male intruder and pink cells that responded to the female intruder. And what you can see are these are largely non-overlapping cells. They either respond to the male or the female intruder. They then took the activity of these cells and looked to see if they could decode the animal's behavior based solely on the activity of the cells. And so they found that indeed they could do this. They were able to decode male and female directed behaviors like attacking, sniffing, mounting, again, based solely on the neural activity. Another interesting finding was that if they took socially naive mice and exposed them to the male and female intruder, these cells actually did not discriminate. So there was no divergence of male and female ensembles, as you can see here in the top panel. However, as they introduced the males to the male and female intruders over days, they saw that the neuronal ensemble started to diverge. And this was a really interesting finding because until then they thought that this neural circuit was very hardwired, but they actually found that it required learning in order to have these neural circuits diverge into the male and female encoding. So I'll next talk about our Invoke microscope. So this microscope is similar to the Envista, but we added on capabilities for optogenetic modulation of the same field of view as the calcium imaging. So this enables a number of different applications that are outlined here. So the first is interregional functional connectivity. So this is where we want to image postsynaptic cells in a particular brain region and then express the opsin in presynaptic inputs. And then we can modulate those inputs to see how they are causally linked to both the neural circuit dynamics as well as behavior. Second application that Invoke enables is looking at functional cell type mapping. And here we can, for example, inhibit the same cells that we are recording from to see what the causal effect is of these cells on a particular behavior. And then the third application that Invoke enables is microcircuit functioning. So this is where the opsin and indicator are in different cell types within the same field of view. So an example of this would be if the opsin is in interneurons and the indicator is in projection neurons, and you want to see the causal link between this neural circuit on behavior. So one type of research that's enabled by Invoke is connectivity mapping. So this study was published from Anatol Kreitzer's lab. Here, they were interested in the dorsal striatum and parvalbumin interneurons. And they really wanted to see what's the functional connectivity of this circuit in freely behaving animals. So they recorded from medium spiny neurons, which are the principal neurons of the striatum, and they expressed halorodopsin in parvalbumin neurons within that brain region. They then inhibited parvalbumin neurons and recorded from the MSNs, the medium spiny neurons. And if you can look at the example traces here, what they found is that when they turn the optogenetic light on, which is then inhibiting parvalbumin neurons, they see an increase in activity of medium spiny neurons. So this confirms that these parvalbumin neurons are indeed functionally connected to these medium spiny neurons, and by inhibiting the parvalbumin neurons, they're actually disinhibiting the medium spiny neurons. So that's also shown here on the right on this raster plot, where you can see an increase in activity when the optogenetic light is turned on. So before I go over the Mira platform, I want to just give a little bit more background on Inscopix as a company. So we are really motivated to create an end-to-end solution for our customers in order to allow them to get to their data and their research questions as soon as possible. So to enable this goal, we started with a surgical workflow and designed a couple of products that enable you to have a much more streamlined surgical approach. So the first are these RTI virus, which stands for Ready to Image Virus. So these are viruses that we provide that we have tested and validated in our lab at Inscopix, and we have determine which construct, which dilution, which serotype works best for particular brain regions and particular species. We also have what we're calling express probes, which is where the Grin lens, the docking station, and the virus is all combined into one step. So this is an incredible way to streamline the workflow. It's a one-step surgery that gets you to your imaging much faster than the traditional approach, which is virus injection, then lens implantation, then baseplate installation. So of course we have our instrumentation, our Envista and Invoke microscopes, and I showed earlier how these microscopes can really enable scientific research. And then we have a really nice data analytics platform that's been validated internally, and it's very intuitive to use to take you from your raw calcium movie through all the way up to extracted cells so that you can see your traces and events for particular neurons that you can then align to specific behavioral events. In addition to this, we also have a really robust scientific support infrastructure. So we have field scientific consultants who are PhD level neuroscientists that come out to customer labs and work directly with customers to make them successful with their particular experiment. We also have workshops that we host on site at our headquarters where we can show firsthand what the surgeries look like and what the imaging looks like. We also have a knowledge base on our website that has a lot of protocols, application notes, case studies, a lot of really great information there as well. Okay, so moving on to the rest of the segment, which will cover the MIRA platform. So MIRA, again, stands for Multimodal Image Registration and Analysis. So this is a fully integrated solution that helps to facilitate the incorporation of data sets collected with the Inscopix miniscopes to data sets collected from benchtop high-resolution laser scanning microscopes, and I'll probably refer to these as LSMs going forward. So the MIRA platform has two different components. The first is a hardware component, which are these two adapters. So we have a multi-photon adapter and a confocal adapter. And the goal of these adapters are to align the two imaging modalities to ensure that we're imaging from the same focal plane, so the same exact brain region. So the way that works for the multi-photon adapter is we have our Envista miniscope that goes here, and then this interfaces with the two photon. In the case of the confocal, the miniscope goes here, and this connects to the confocal. And what this does in combination with the calibration slide is that this brings the two microscope modalities to the same exact focal plane in Z. And so that's really the first step of our ultimate goal, which is to identify the same exact cells that are imaged with both modalities. So once we get our data collected with the adapter, we know that we're in the same Z plane. We then use an analysis tool to align and register the images so that we can, again, identify the same exact cell image between the modalities. So I'll go into detail for each of these steps in a minute. So the reason why we wanted to work on this MIRA platform is because we really wanted to combine the power of free behavior neural circuit imaging with the benefits of high-resolution multicolor imaging. So of course, with free behavior imaging, we are able to investigate neural circuit dynamics and naturalistic behaviors. And then we also, within the same experiment, want to add on the information that we can get from high-resolution multicolor imaging. So the general workflow of the MIRA solution is first we image our neural circuits with the Envista miniscope during free behavior. We then use those two adapters that I mentioned, either the multi-photon or the confocal adapter, to align the fields of view between both modalities. We then do our head-fixed imaging under the LSM to get the high-resolution multicolor information. And then we use our analysis platform to align and register the cells to identify which cell is the same cell between both modalities. So this enables a number of applications. So the first two are dual color imaging applications. So here we want to image our neural circuit during free behavior, and then we want to identify a subpopulation of cells that are tagged in red. These can either be projection-specific neurons in the projection identity application shown here on the left, or this could be where the red indicator is in a genetically defined population of cells shown here in the genetic identity application. The third application is behavioral state identity. So this is where we are interested in looking at the differences in neural circuit activity under different behavioral states. So of course, a lot of behaviors are really well suited for free behavior. We want to really understand how these neural circuits are involved in naturalistic behavior. However, there are some behaviors that are more well suited for head fixation. So these could be, for example, tasks where we want the mouse to be able to pay attention and orient to a specific area in space. And so in that case, we want to compare that particular neural circuit under different behavioral states. And then the last application is where we want to combine neural circuit imaging with non-neuronal imaging. So for example, if we're interested to see how neural circuits interact with things like microglia or astrocytes or pathology such as plaques, we can image those using a red indicator under the LSM. So I'll next walk through a specific application that we have validated in our lab at Inscopix. This was a project that was in collaboration with Zeiss. So we combined our Envista imaging with the Zeiss ARIScan imaging in order to do this projection identity application, which is again this first application shown here where we want to image a subpopulation of cells that project to a different brain region. So specifically what we wanted to do was image medial prefrontal cortical neurons during free behavior with Envista and co-register those cells that are projecting to the contralateral hemisphere in red with the Zeiss ARIScan. So to enable that, we have a workflow shown here. So we first do our PFC free behavior GCamp imaging with Envista. We then do the field of view alignment using the confocal adapter. This aligns the Envista field of view with the Zeiss ARIScan field of view. We then image the GCamp under the ARIScan, and then we image the static red under the ARIScan. So this static red, in this case, is a retrograde TD tomato that was injected in the contralateral hemisphere. And so what you can see here on the right, hopefully, are some yellow cells that indicate cells in the PFC that are projecting to the contralateral hemisphere. And then our ultimate goal is to analyze which GCamp neurons we image during free behavior that are co-expressing the static red indicator and thus are the contralateral projectors. So I'll walk through each of those individual steps now. So again, our first step is to do our Envista free behavior imaging. So here we're recording from prefrontal cortex. You can see the mouse here is engaged in an open field behavior, and that's synchronized to the calcium imaging shown below. So once we do our free behavior imaging, we then want to find that same field of view under the Zeiss ARIScan. So to do that, we align the two fields of view with the confocal adapter. So again, here the Envista is sitting on top, and then this bottom interfaces with the camera port of the ARIScan. We use a calibration slide that contains a single layer of 15 micron fluorescent microspheres, and we use those microspheres to align the two microscopes so that we know that we're imaging from the same focal plane. So once the two microscopes are aligned, we can then switch over to the ARIScan imaging and know that we are imaging from the same focal plane that we were imaging from with the Envista. So that's shown here. Here you can see the animal is head fixed on a running wheel, and we're imaging from the same prefrontal cortical circuit. So I should also mention, sorry, here we are imaging two things. One, we're imaging the GCamp activity, and then we're also imaging the static red channel to get that information about which cells are contralateral projectors. So once we have our GCamp imaging from both modalities, we then take it through our standard processing pipeline, which is outlined here. So the first step is to pre-process the movie, and oftentimes we want to downsample in space, and we also want to crop. We then want to correct for any brain motion that may be evident in the movies. We then want to normalize the fluorescence so we can look at changes in fluorescence over time. And then we want to extract our cells. So once we do that for both the INVISTA functional movie and the ARIScan functional movie, we're left with these two functional cell maps. So every green circle here is a GCamp cell that was extracted from the calcium movie of both modalities. So again, because we used our confocal adapter, we know that we're imaging from the same brain region and the same field of view, but what we now are left with are these two cell maps that are nearly impossible to identify the same cell between both modalities. So it's very difficult to pick out any similar cell between the two modalities, and that's because we need to take it through further processing to correct for a number of different things. For example, we need to flip and rotate the field of view so that they match. We need to correct for different pixel numbers and sizes of the images. We need to correct for optical aberrations in XY that are due to different, the way that the two different microscopes image. And then we also need to correct for the achromatic aberrations when we want to add on the red channel. So I'll walk you through how we go through each of these corrections in order to have our final goal, which is to overlay the two cell maps. So one thing that we take advantage of when we're trying to match the two cell maps is actually the structural information from the images. And so when we look at the structural information, we can actually start to see blood vessels pop out. And these are really useful to help us align the two images. So to get this structural information with the INVISTA movie, what we do is we maximum project the movie to a single frame, which is shown here. It's also important to note that our depth of focus with the INVISTA is 50 microns. So we want to match that depth of focus with the ARIScan. So to do that, we take a Z-stack where the zero plane is that same plane that we identified from the adapter. We then take sections up to 25 microns on either side, which in total equals 50 microns to match the depth of focus of INVISTA. We then project that image into a single image. We then rotate and flip the INVISTA image so that it orients the same as the ARIScan image. So now you can start to see some similar landmarks and structural information between the two modalities, such as this cell here, this blood vessel here. And what we do is we then take a look at the two images. We identify similar patterns between the two images, typically about 5 to 7. And then we use that structural information to then warp the INVISTA image to match the ARIScan. So here we're registering the two structures together so that we can then overlay the INVISTA image with the ARIScan image shown here on the right. So INVISTA is in green, ARIScan is in magenta. So once we get this transformation matrix from the structure, we can then just directly apply that transformation matrix to the cell map of the INVISTA image. And so when we do that, now it's very clear which cell is the same from both modalities. So again, here we have our ARIScan cell map in magenta. This actually was not changed throughout the analysis. That was the first image that we saw earlier. But here in green is our INVISTA cell map that was transformed to match the ARIScan cell map. And so you can see green cells that were from INVISTA, magenta cells that were identified from the ARIScan, and white cells that are the same cells identified with both modalities. So this would be an application where you want to image the same neural circuit under different behavioral states. But for this particular application, we wanted to take it that extra step of looking at the projection identity application. So here we actually want to overlay the INVISTA cell map with the red structure information from the ARIEScan. So we took a Z-stack in the red channel. We then segmented out the red cells, and then it was pretty straightforward for us to then take the transformed INVISTA cell map and directly overlay it with the ARIEScan red cell map, which were the red segmented cells from the structure information. And here, what we now see is the green cell map overlaid with the red ARIEScan cell map. So all of these red cells here are contralateral prefrontal cortical projectors, and the yellow cells are cells that also co-express GCAMP. So all of the yellow cells here are GCAMP cells that we identified during free behavior that are also co-expressing the static red and thus projecting to the contralateral hemisphere. So we identified matching cells using a normalized cross-correlation or NCC matrix. We then have defined thresholds for cells that are either co-expressing the red indicator or not co-expressing the red indicator, and these can be user-tuned. And so here's our a couple of example traces where green traces here correspond to cells, for example, cell number 4 that's not co-expressing the static red. And then an example for example here, cell number 2, which is right here, that is actually co-expressing the static red. So this would be an example of a contralateral prefrontal cortical projector. Okay, so to summarize, we showed in the first half that the Inscopix platform enables optical imaging and manipulation of large-scale neural activity in brain circuits during behavior. And then with the Mira platform, we can take all of the benefits of that neural circuit dynamic information that we get during naturalistic behavior and add on the extra information that we get from the LSM to enable these applications such as projection identity, genetic identity, looking to see how cells respond in different behavioral states, as well as combining neural circuit activity with non-neuronal imaging. And with that, I will take any questions.
Perfect. Thank you so much, Alice. This was such a great presentation. It's actually super exciting for all of us to finally be sharing more about the Mira platform with everyone. And so, yeah, we can get started with a little Q&A session here. We do have several questions that have already come in to begin with. So maybe we can start with this. Alice, if you want to take a look at the Q&A box, if it helps to read also.
Okay.
The first question is from Ryuichi Nakajima, who just seems like wants clarification on what we mean by behavioral state identity, and how you can differentiate between the two GCAMP signals.
Yeah, so with behavioral state identity, this is where we want to compare neural circuit dynamics with a behavior that requires naturalistic behavior with a behavior that requires head fixation. So the naturalistic behavior data is collected with the Inscopix Miniscope, either INVISTA or INVOKE, and the head fix behavior is acquired with the LSM. So these are two separate cell sets that are acquired during different times. So the way we can differentiate the two GCAMP signals is just due to the difference in time. They're completely different movies. We process them independently. We can understand the dynamics during free behavior with our INVISTA imaging, the dynamics during head fixation with the LSM imaging. And the goal of the Mira is to, after we analyze those two data sets independently, we want to know which cell is the same between both modalities. And so we do our registration to identify the same cell. So hopefully that answered the question.
Yeah, I hope so. Please, anybody that is asking questions, if something's not clear, please follow up with in the Q&A box with follow-up questions. Okay, so the next question is from Sean Lim. He's asking if the co-localization analysis requires cell segmentation within the Inscopix data processing software and whether it's compatible with other cell segmentation programs like Command.
Yeah, so that's a great question. Actually, this data that I showed here today was analyzed with CNMFE. So, yeah, Cayman's definitely supported with our processing software, although it's not in the GUI, it's in the API. So we support both PCA, ICA, and CNMFE. If you want to do any cell extraction outside of the Inscopix platform, that is not currently supported.
Okay, hopefully that answers Sean's question. Okay, so we have a question from one of our customers who's Cody Siciliano at Vanderbilt, and he's asking if we can explain a bit more about the transformation to align the images, if it's linear, and whether there's any concern about the possibility of non-linear magnification of cells imaged on the Envista, which are slightly above or below the focal plane.
Yeah, so I guess this maybe is a two-part question. So the first part, the exact type of registration we're doing is non-rigid registration. I guess maybe the question about magnification of cells, so that's actually not a concern with our Envista 3 and Invoke 2. So I should clarify that the Mira platform is only compatible with Envista 3 and Invoke 2. And with those systems, we do not have the if we go above and below the focal plane, we actually are not changing magnification. The magnification stays the same. So we don't have any concern about changes in magnification. Yeah, and then I guess, for cells that are above or below the focal plane, I mean, again, here let me come back to this. Sorry. We are matching the depth of focus between the two modalities by, so here our Envista imaging is 50 microns with the ARIScan, with the ARIScan we're collecting a Z-stack of 50 microns to match that focal plane.
Mm-hmm. Yeah, okay. Hopefully that helps. Cody, please let us know if you need more information. Happy to talk offline with anyone too. We have obviously our contact information on Alice's deck. You can find us on the website as well. Okay, let's see. So I guess a couple more questions on just the analysis part of it. There's somebody asking how to get reliable structural landmark matching, if this is an automated or a manual process where the two images might look different by eye.
Yeah, so this is definitely an important step, is to make sure that we do have reliable landmarks. Currently, the Mira platform is supported with the 1 millimeter by 4 millimeter Gren lenses. And so these are pretty large diameter Gren lenses. And what we've seen is that if we optimize our surgical approach, we see actually really clear fields of view. So that's definitely an important step to optimize during the surgery. And that's something that our field scientific consultants can help with. And then, so yeah, I would say once your surgery is optimized, you should not run into too many issues of seeing clear landmarks. So hopefully that answered the question.
Mm-hmm. Yeah. Okay. So Amol Bhandare is asking whether during the LSM imaging part of it, do we acquire the Envista signal to have both imaging done at the same time? I guess just the workflow, a bit.
Yeah, so the Envista imaging is done separately from the LSM imaging. So we really want to make sure that we preserve the free behavior capability of the Envista system. So that's done, for example, on day one, you do your free behavior imaging, and then on a separate day or later in the day, you can then connect your animal to the head fixation setup and do your LSM imaging. So these are two separate imaging sessions. And the purpose of having the miniscope connected to the adapter is really just for field of view alignment.
Right. Okay. So, yeah, I guess this question from Xiaorong Ma, I hope I'm saying that correctly, is also about just the process of co-registration. Says, from the mirror example, I saw some green and red cells partially overlap. Are they different cells or the same cell?
Yeah, that's a great question. Let me get back to that slide. Okay, so yeah, maybe they would be asking about this cell in particular here, where
I think so.
maybe it's like a 40% overlap. So these are user-defined thresholds, so you can make these as loose or conservative as you want. So if you want, maximum spatial overlap, you can set that. And you could, do some experiments to determine what you want that threshold to be. So that's totally up to the user to set that threshold. And so again, if you want to be super confident, you can set it very high, like 0.9, and then that will only give you the cells that are co-localized that have a very, very high spatial overlap.
Mm-hmm. Okay, yeah, hopefully that helps Xiaorong. Let's see. Okay, so I think Cody is following up with another clarification, asking if we're attempting to co-register with the 2P and therefore using a much longer wavelength than the INVISTA, how will the chromatic aberration introduced by the grain lens be dealt with?
Yeah, that's a great question. So I actually didn't cover this in my talk, but I do have a slide just on the chromatic aberrations. This is actually with the Zeiss Aeroscan confocal, but I can maybe talk about this and then address your specific question about the 2-photon. So we know that Grin lenses introduce chromatic aberrations. So we wanted to measure what that is first with the confocal. So we had microspheres that contain both green and red dye. And what we found is that on the confocal, if we image the green beads and then switch over to the red channel, they're not there. They're not in the same focal plane. We actually had to move the stage 35 microns to bring those red beads into focus. And then that overlaid really nicely. So with the confocal, we see that this red-green shift is 35 microns, and this is to be expected because we're imaging with two different wavelengths. With the 2-photon, with your red and oops, what did I do? Sorry. With the 2-photon, your red and green typically are imaged with the same laser, so you don't necessarily have to worry about this particular chromatic aberration. With the longer lenses, it definitely gets more severe, and so that's why currently we're only supporting the 1x4 lens. We're looking into other options to enable deeper brain regions with longer lenses. I think that maybe you're also asking about, with the 2-photon, you have IR lasers, so that introduces another aberration. And that's something that we're going to look at with not beads in particular, but in a animal where we have, instead of a dynamic green indicator, a static green indicator, and then we can image that animal between the two modalities as sort of a ground truth experiment where we know that the cell is on with both modalities, and so we should be able to get a better handle of the green to IR chromatic aberration.
Yeah,
but yeah, I guess you're specifically referring to excitation rather than emission. So yeah, I guess in the case with 2-photon, you're using the same laser for red and green.
Okay, great. So here's another question. So yeah, specifically regarding the co-registration analysis workflow. The question is why we transform the Inscopix image to match the LSM image, and whether it can be done the other way around.
Yeah, That's a great question. So I'll go back to that slide here. So yeah, delayed. So yeah, we are warping the InVista image to match the ARIScan image. The reason why we did that is because we want to preserve the high resolution information, in this case with the ARIScan. And so, for example, if you want to start looking at high resolution structural information, you have that resolution preserved. The only downside to this, our current approach, is that we have to upsample the InVista movie, which just adds a little bit of file size. But yeah, we can totally support the opposite way where you want to transform the LSM image to match the InVista image. That's definitely supported.
Okay, great. Another question is, what types of, or what are the requirements? I guess maybe we can address the microscopes that we are compatible with.
Yeah, sure.
Yeah.
So with the confocal adapter, we're compatible with the Zeiss ARIScan 880 and 980 model. With the multi-photon adapter, we're compatible with, there's, I guess, a number of requirements. One is that the multi-photon is capable of live mouse imaging. So we want to make sure it's an upright microscope. It has enough space to fit a mouse on a running wheel or whatever head fixation setup you have. We also want to make sure it's fast enough for GCam imaging. So it has, for example, a resonance scanner that allows for high frame rate imaging in a large field of view. Other than that, I think if this person wants to contact support at Inscopix.com, we can work with them on their particular LSM model to make sure it's compatible.
Yeah, I think this question was by Didi Mamaligas. Didi, please, like Alice said, contact us if you need more information.
Yeah.
Okay. We have a little more time. If people wanted to go ahead and submit more queries, we're happy to answer. There's a couple more that we can talk about in the meantime. So it's, I think one of our most commonly asked questions is whether we can use Mira to co-register fluorophores other than RFPs.
Yeah, that's a great question. So I guess there's a number of factors to consider. One is just whether the LSM can spectrally separate that particular color. So we know that we want to have GCamp or GFP imaging as one indicator, and then you would want to make sure that your second indicator is spectrally separated from GCamp. So this would be a question of how your LSM is configured. And then once you can confirm whether the indicators are spectrally separated, I think the question really depends on if you're imaging on a multi-photon, which may not have the chromatic aberration issue that we would see with the confocal, again, just depending on which lasers you're using. So it's a bit of a complicated question. So I would definitely encourage this person to email support at Inscopix.com. We can talk through your specific application. We can talk through what wavelengths you're interested in, what lasers you would be using to excite those, and then we can, once we have a handle on the chromatic aberration and just a better understanding of the application, we can assess whether it's feasible.
Right. Okay. So another question is if somebody is expressing an opsin that is tagged or like co-expresses a static fluorescent protein, are there limitations to the wavelengths that will be compatible?
Yeah, that's an interesting question. I think, that could be maybe like a specific application of Invoke where they want to do their free behavior imaging and, for example, inhibit a subpopulation of cells, and they actually want to know which cell they inhibited. Typically the opsins that we support are tagged with a static red fluorescent protein, whether that's TD tomato or mCherry. So just, from the spectra characteristics, we can certainly image that on the LSM. I think the questions with opsins in particular we have to consider that the opsins are expressed on the membrane, and a lot of, for example, here when we're looking at this TD tomato, it's expressed in the cytosol, so it's just much easier to image. When it's expressed on the membrane, sometimes it can be hard to, extract the cell. So I think, again, that's one of those questions where I definitely encourage this person to contact support, send us some images of what the static protein expression looks like under their LSM, and we can see if we could segment those cells out that are expressing the red protein.
Okay, yeah. All right, so maybe there's one more question and we might wrap up with that if there are no more. And this is just a validation question. We're being asked if we can confirm with histology the cells that were imaged in vivo.
Yeah, that's a great question. So I think this person's asking, can we take in vivo imaging from the INVISTA and then perhaps register it with histology? So the Mira platform only supports registration between in vivo and in vivo, whether that's confocal or multiphoton. There was actually a paper published earlier this year from one of our customers in Journal of Neuroscience Methods, and they actually did just that. So they registered INVISTA imaging to histology. They use a lot of the same algorithms that we used here and methods. So I would definitely encourage that person to look at that paper and go through the methods because, that is something that has, at least the proof of principle has been shown. And it's a really cool paper. Once you can register to histology, you open up a lot of really cool applications.
Right. That is true. Okay. All right. I think we might be at the end of our list of questions. Thank you so much, Alice. This was a very engaging discussion on what we're hoping will be a very helpful and insightful platform for everybody to use in addition to our miniscopes. Okay, and then like Alice and I have mentioned several times now, please feel free to contact us via our support contacts or just on our Inscopix website. We have a contact us page, so you can just click over there and submit questions too. And we're just looking forward to all the amazing science that might come out of everybody using the Mira platform in the near future. And thank you so much again, Alice. Thank you everyone for attending and participating with questions. I hope everybody has a wonderful rest of the day. Bye, everyone.